welcome to my third episode of this science podcast. Today I'm delighted to be joined by Professor Dame K. Davies, who is Dr. Lee's Professor of Anatomy at the University of Oxford, who set up the MRC Functional Genomics Unit and co-founded the Oxford Centre of Gene Function, as well as being the co-director of the MDUK Oxford Neuromuscular Centre and a Fellow of the Royal Society. I hope you enjoy.
Fun? Okay, recording. So I began my career as a chemist, and at Oxford I did chemistry, and fortunately for me, the last year you could choose any area of research that you wanted to do. So I chose biochemistry, and so from then on I stayed in the biological sciences and moved into genetics, and the rest is history, because genetics was so exciting in the early 1980s, and so my career started there really. And I was in France for a couple of years and then came back to London to start the genetics of Duchenne muscular dystrophy, DMD, and then I moved back to Oxford to continue that work. And since then have moved, since the gene was cloned, I've been very much focused on developing a therapy.
That's fascinating. And so in your report of the heritable human genome editing, what ethical aspects of the technology arose and what conclusions did you make from this?
Okay, well, we weren't charged with looking at the deep ethical and social issues, only those that, because there's a partner report coming from the WHO, which is looking at the governance and ethics, but obviously we couldn't avoid the ethical issue because they're very important. So the major ethical issue is how do you make sure that the babies that are born after HGGE are followed through so that there aren't any adverse effects? So looking after that sort of family and doing follow through was very important. So that's one ethical issue. The other ethical issue is to balance the risk that you would produce by manipulating embryos versus the benefit. And that's why we deem that it really has to be very, very safe before the benefits would outweigh the risks. And for now, therefore, the conclusion was that you shouldn't do human germline gene editing.
Yeah, it would have to be the very last resort if there was no other option of reproduction, because you can't do prenatal testing or pre-implantation genetic testing, and they have no other option. Also, this would reduce the risk of any rogue scientists, if you like.
Well, we would hope that since this was an international commission covering 18 countries, including China, where the first babies were produced, that there was an international agreement that everybody should coordinate regularly on this. And what had happened when those babies were born in 2018 is there wasn't that international dialogue. Now, you can never stop rogue science completely, certainly minimize the risk of it happening. And I think what we've done is substantially minimize the risk of it happening, because internationally people would know that it has been unsafe at the moment and we shouldn't be doing it.
Absolutely. So what disorders would qualify for this technology?
If it was safe, then it would be those rare conditions where there are no other options, where a homozygote affected sickle cell patient married another sickle cell patient, or a cystic fibrosis patient married another cystic fibrosis patient. Now, those are relatively rare, but having said that, there are family groups which join together to tackle disease. So actually, these people do meet each other and may get married and may wish to have children. So this would give them that option. So those would be, in cystic fibrosis, that would be the Northern Hemisphere, UK in particular, but perhaps sickle cell anemia is the one. And that's inbred populations in places like Africa and India. And there are cases of that in the States. I mean, sickle cell anemia is not as rare as you might think.
Okay. In heritable human genome editing, how could you ensure that when you're reverting a mutation to a naturally occurring allele, for example, you don't take out a control region on the adjacent gene or something like that?
Yeah, that's very important, on target specificity. In fact, that did happen in the experiment that was carried out that shouldn't have been carried out in China on those babies. And that's why we say that you have to We will only, in the first instance, you have to take those cases where there's a single base mutation and you're taking it back to what is normally seen in the population. And you're not trying to do anything too complicated, and that you have to check on target, which is what you just mentioned, as well as off target effects into account, because it does naturally scramble whenever you interfere. And we have to check that in human embryos, not just in mice, because mouse embryos don't react in exactly the same way as human embryos do.
No.
That is also extremely important.
Okay. So how do you think that somatic cell editing and gene therapy, the cost may be decreased in future years, because right now it's extremely expensive?
Yep, it is £1 million a patient. And a lot of that is because the vector production is very expensive. But technology is coming down the pipeline to increase that, the efficacy of that, at least tenfold. So the price will come down. There's no question. And there's a huge push from the industry to do exactly that, to make it more routine. You can take the humans out of it, make it more automatic, pipeline and safer. So it's going to take some years to get there, but I think there's no question that gene therapy, those vectors will be cheaper and safer.
That's brilliant, and that is music to many families' ears, I'm sure.
Oh, yes.
So we're going to move on to a bit about your work on Duchenne muscular dystrophy now. So how may CRISPR technology be used for patients with this disease?
Two ways of doing it. One is that 10% of patients have point mutations. So you could use CRISPR by injecting the Cas9 and the corrected sequence into the patients. And this has been done in mouse embryos with point mutations, and it works. The problem is the risk and and you'd have to go in early. So I think there are major safety concerns, because not only an AAV that have to have sequence in it, you'd also have to have all the machinery for CRISPR delivered by AAV. So I think that's a long way off. Eric Olson's group have also, a lot of patients have developed exon skipping CRISPR oligonucleotides, and they again work very well in the mouse, because a lot of patients have the same portion of the gene missing. You could use that CRISPR technology, 50, 60% of the patients. But again, problem, you've got to get the Cas machinery, Cas9 CRISPR machinery in, and that's going to be still an immunological challenge if you have to do it systemically very early on. Definitely.
So dystrophin is associated with the cytoplasmic face and muscle fiber membranes, and its C-terminal domain, the COOH, is thought to mediate membrane attachment. However, the precise function of dystrophin is unknown. So what kinds of studies could reveal this?
I think more biochemistry, where you look at the signaling molecules. And we already know that it's not just a structural molecule that connects the outside extracellular matrix to the inside of the muscle cell, that there are signaling molecules that bind to the membrane. So we need much more biochemistry. To put that. Some very good biochemistry has already been done, but we probably need more, and a few more gene knockouts to show that some of these molecules are important in the function of dystrophin.
Absolutely. May you elaborate on your work on the upregulation of utrophin in treating DMD?
Okay, utrophin's a gene that we discovered when we were looking for dystrophin, and it turns out to be on chromosome 6 and not the X chromosome. And you can cure a mouse by introducing more utrophin in the absence of dystrophin, and it substitutes completely. Recently, a group in the States, Hansel Stedman's group, have shown that if you do that in the dog, which is a much more bigger muscle animal, you can do the same thing. You can rescue it by increasing the levels of utrophin. So what we've done is set up cell lines so that we can monitor drugs that might be able to increase the level of utrophin in those muscle cells. And azutamid was the first drug we took to the clinic, and it failed in the clinical trial because of the metabolism of the drug. And we now need to find another drug which works like azutamid, but doesn't have all of the bad pharmacology which caused it to fail.
So it's on the brink of success.
We hope, yes. And we've just got more funding for that. Absolutely.
Okay. So how is the raised levels of 2+ calcium ions in dystrophin deficient muscle an important cause of the pathophysiological processes leading to cell death? And how may increased membrane permeability also relate to this?
It's because the calcium leaks in and activates proteases, which then degrade lots of protein in the muscle, and you just get muscle breakdown.
Right.
That's a very simplistic view, but there are lots of different calcium channels. But the most important mechanism is this activation of proteases. So that's why people are working on sealing the membrane to prevent that from happening.
Okay.
Which is laminin 111 or the biglycan. You may not have heard of these, but these are molecules that block those gaps in the membrane. And so doing so, they stop this infiltration of calcium.
That's brilliant. What evidence do you have that implicates oxidative damage early in the dystrophic process?
This is a fairly controversial area. Some people feel that this is really important and others feel it is less important. So your question is, what's the evidence? Well, I think I'd just say it's controversial. If you cross a mouse which is deficient in oxidative stress with the MDX mouse model, which doesn't have dystrophin, you get a slight decrease, you get a slight worsening of the phenotype, but it's not as dramatic as you might think if oxidative stress played a central role. Having said that, drugs that control oxidative stress do appear to have some benefit in patients, but it's only a slight benefit.
Okay. Why is the idea that MDX muscle in mice regenerates less well than normal due to sustained high satellite cell turnover? Why is this uncertain?
I don't think it is uncertain. I think it's definitely true that the MDX does regenerate much more efficiently. And what happens in humans is that you get an infiltration of collagen, which then just prevents, there's no matrix left no room left to regenerate the muscle effectively. And we know, for example, that utrophin is a part of that regeneration process. So if you take an MDX mouse and cross it with a mouse that doesn't have any utrophin, you get a very severe phenotype. So I think that tells you that it's the regenerative process that's so important, that works well in MDX, but doesn't work so well in man.
Absolutely. My last question is, dystrophin and utrophin are components of the postsynaptic apparatus in many CNS neurons, and particularly the cerebral cortex. Therefore, your research may serve as a model not only to study potential therapies for DMD, but also provide clear insights into subtle neurogenic components in the pathology of the muscle dystrophies as a whole. So what further studies is this research area lacking so far in achieving this goal?
I think that we need more studies on the function of dystrophin in the brain. And there are different isoforms of dystrophin that are expressed in the brain. And we certainly need some more biochemistry of different brain areas and functional studies in utrophin deficient mice as well. And those studies have not been done. People are very much focused on trying to improve the quality of life of DMD patients by clinically benefiting their muscle. But of course, a lot of them do have mental impairment. And so the next stage will be to identify what that function of dystrophin is and what might be done to help these patients in that regard. Yeah.
Okay, wonderful. Thank you so much for joining me today. Do you have any other areas you'd like to expand on, or any questions you'd like to ask me?
No, it's just been terrific to interact with you, and all the best for the future.
Thank you very much, and thank you for listening, everyone.